Hi everyone, today's lesson is about antibodies in medicine. In a moment, pause the video and think about what monoclonal means. So mono means one, like a monobrow is one eyebrow, or a monocle is one eyepiece. That leaves clonal, and we should all know what a clone is. So if I was to say monoclonal antibodies, it would mean that all of the antibodies were clones of just one antibody. Today's learning objectives are on the board. By the end of today, you should be able to explain how monoclonal antibodies are produced, describe the uses of monoclonal antibodies, and evaluate the issues surrounding the use of monoclonal antibodies. First, we will recap antibodies. Antibodies are proteins that are produced by B cells. They are specific to particular antigens due to their variable regions. This means that they will only bind to antigens that are complementary to their variable regions. In a moment, pause the video and have a go at labelling the antibody on the right. So this green region at the top is the variable region. The next label down is pointing to the light chain. The label below this points to the disulfide bridge, and the final label points to the heavy chain. Give yourself a tick for any you got right and correct any you got wrong. The first learning objective is to explain how monoclonal antibodies are produced. In basic terms, monoclonal antibodies are produced from a single group of genetically identical B cells. Monoclonal antibodies can be made that bind to any substance. This could include cells or antigens, among many other substances. We call this substance an epitope. The first step is to inject an animal, for example a mouse, with the specific epitope of interest. B cells in the mouse produce a single type of antibody that are specific to the epitope. The B cells are isolated from the animal's spleen and mix with myeloma cells, a type of cancer cell that grows continuously. The cells are fused together chemically to form a cell called a hybridoma. The hybridoma cells are placed in a growth medium and allowed to multiply. They then produce monoclonal antibodies. The monoclonal antibodies are isolated and then purified. Now we're going to look at some uses of monoclonal antibodies. The first use is to target drugs to cancer cells. Cancer cells have specific antigens on their surface called tumour markers. Monoclonal antibodies are produced that bind to these. Anti-cancer drugs can be attached to the monoclonal antibody. This enables the drugs to be specifically targeted to cancer cells. This is really important because cancer drugs have lots of side effects, so we don't want them to accumulate in other areas of the body. The second use is in medical diagnosis. You are all probably familiar with one particular use of monoclonal antibodies in medical diagnosis. I am, of course, talking about COVID-19 antibody tests. However, the example you will learn about today is pregnancy testing. Pregnancy tests detect the hormone human chorionic gonadotropin, or HCG, in women's urine. The application area of the pregnancy test contains antibodies for HCG, which are bound to a blue-coloured bead. If HCG is present in the urine sample, it will bind to the antibody, forming an antigen-antibody complex. Urine moves up the strip, carrying the antibody and beads with it. There is a test strip at the end, which contains immobilized antibodies for HCG. Immobilized means that the antibodies are stuck there. If the blue HCG antigen-antibody complexes are present, then HCG will bind to the immobilized antibody and the strip will turn blue. This means the woman is pregnant. Here's an exam-style question for you to answer. If you feel confident, try it without your notes. If you feel you need a little more support, use your notes to help you. Pause the video now and have a go. Please use a different coloured pen to mark your work. For the first question, you get 1 mark for saying it is a molecule or a protein, and another mark for saying it stimulates an immune response. For the second question, you get 1 mark for saying they have a complementary shape, and a second mark for saying that this allows them to bind to the specific antigen. Give yourself a total mark out of 4. In a minute, I would like you to read the material on the slide and summarise it in the table, with one column for the benefits of using monoclonal antibodies and one column for the limitations of using monoclonal antibodies. Pause the video now and create your table. Finally, here are some exam-style questions I would like you to have a look at for homework. See you next week!